So I'm Erin Edwards. I am the head of services at Visicall. Visicall is a contract research organization. We specialize in a number of different assays that include imaging as one of the primary endpoints. So these include running in vitro assays, cell-based assays, 2D or 3D cell culture models, where we'll work with clients to evaluate their compounds on those 2D or 3D cell culture models. We'll take them through a number of different endpoints, many of which include imaging. And this is where it gets sort of interesting. We'll take in some other projects from clients who have already generated 3D cell culture models or other tissues and label them in 3D, pull out any sort of quantitative insights by complex analysis of those 3D tissues and cell culture models, and deliver a report to the client in order to drive forward their drug discovery programs. For many of our projects, we use the Thermo CellInsight CX7 LZR high-content confocal imaging platform. It's a great system because it allows us to essentially perform confocal microscopy on a number of samples in a much shorter amount of time than if you were using standard upright confocal microscopy. Confocal is super important when you're trying to visualize 3D cell culture models or 3D tissues because it really allows you to get deeper into those tissues and make sure you're actually analyzing the entire tissue rather than just the first couple of layers. So in many of our projects, we also use clearing reagents, in particular the CytoVista reagents. These allow us to clear the 3D cell culture models or more complex tissues such that we can actually image throughout the entirety of those 3D cell culture models and really get at those quantitative insights that cover the entire 3D cell culture model. If you only image the outside of the cell culture model, it's going to be not the greatest representation of what's actually going on in that tissue. So we really leverage those technologies, the combination of the clearing reagents with the high content imaging platform in order to extract the most important quantitative insights and fully characterize these models. So with many of our assays, the advantage of using the Thermo CellInsight CX7 LZR platform is that it allows us to go much more quickly in the imaging phase. So if you had to go through each individual tissue and image it in confocal microscopy mode, if you're doing that on a single tissue scale, it'll take you hours, maybe days, and require constant interface with the equipment. However, with the Thermo CX7 LZR, we're actually able to accomplish that same amount of imaging in a mere 1 to maybe 3 hours, depending on how many different types of endpoints you're looking at. This especially becomes important when you're talking about doing a high content screen, something where you're trying to get a lot more out of those 3D cell culture models. You might have 4 or 5 different colors, different channels that you're trying to image in, maybe 15 or 20 Z steps in a 96 well plate, maybe a 384 well plate. That would take days for hopefully not a grad student to go through and scan each individual tissue. So the equipment really allows us to do that in a much more high throughput manner. So we work on a lot of different projects at Visicall. We don't really have a particular therapeutic area that we narrow into. Instead, we work in many different areas. So it allows us to get exposure to all of these different therapeutic contexts. Things like immuno-oncology, where we'll look at immune cell invasion into tumor spheroids. Again, clearing and high content confocal microscopy are huge for this assay. It allows us to see how deeply immune cells are actually infiltrating into spheroids and how a client's compounds might affect that ability for those cells to infiltrate.